The immune system is our body's greatest defense against outside invaders such as bacteria and viruses. It has two separate branches, a specific branch and a non-specific branch. The non-specific branch doesn't know what it's actually targeting when it's protecting the body. Instead, it's a bunch of organs that just protect the body against anything that's trying to get in. Skin is one of our body's biggest non-specific organs. It doesn't know what it's protecting against, and it usually keeps out the bacteria and viruses that are trying to get in, but not always. Sometimes they do end up getting into our bodies, and this is when we need help. The specific branch takes over from the non-specific branch when invaders make their way into the body. Its main defense mechanisms are the white blood cells. These target the specific threat in the body. They are dendritic cells, B cells, macrophages, natural killer cells, which are NKCs, and T cells. This specific system works together to attack and destroy these invading bacteria or viruses. First, the macrophages identify that there's an invader in the system. It then engulfs the invader, in our case a bacterium, and destroys it. Next, it releases an antigen that is a specific protein to the invader that helps the cells identify it. This alerts a T cell to come and dock on the antigen. The T cell then relays the message to the B cells. The T cell then takes the antigen and creates a protein that the B cell uses to create antibodies that are specific to the invader. These antibodies are specific to the invader and only the invader. At the same time that these antibodies are being produced, the B cells are also multiplying. The antibodies go out and target the invaders. As they do so, they attach to the invaders and destroy them. This process occurs many times until the body is rid of the invader. The antibodies stay in the body, though, in case the invader comes back. The purpose of performing an ELISA is to determine if someone is infected with a disease. We start with an empty well. Then we add our antigen. The antigen binds to the well walls. We then use a buffer and wash it twice to get rid of the extra unbound proteins. Next, we add a primary antibody. The primary antibody attaches to the antigens, and we do another wash. Third, we add the secondary antibody with an enzyme attached. This antibody attaches to the other antibody. Here we perform another wash. Finally, we add a substrate to the well. If the bacteria is present in the well, then the substrate will react with the enzyme and the solution will turn blue. If the solution turns blue, then that means that the person is positive and is infected with a disease.